I'm going to talk about some science and how some drugs came about, focus on HIV and viral hepatitis, and then about the access. We would never have access if we didn't come up with the great drugs first. And I like this type of meeting that Raymond runs here, where there's a large cross-section of people in the various parts of science and other parts of drug development that come together to talk about the future. This is just 13 years ago, Bill and his daughter, who's here in the room today, came to a conference in Barcelona. And as Raymond mentioned, we all saw Bill Prodrugs all over the world. In the 80s, when I was working for Bristol Myers, first in Syracuse, then in Wallingford, Connecticut, he'd be up in Syracuse in the middle of winter to come consult with us. And of course, once we moved to Wallingford, we're just like 20 miles from his office in New Haven, so I was more often there. To begin off, a talk on HIV and viral hepatitis and where the drugs come from. The most amazing thing is what happened in the beginning. All of you heard Mitch Mitsuya talk yesterday, and he was talking about his work in hepatitis B, but what is his most remarkable work is developing the first screening assay for HIV. AIDS was recognized only in 1981. In 1983, we knew it was HIV. By then, many of us already had successful careers. Raymond was talking to me this morning about some work he did well before this in feline herpes viruses, for instance. So just many things going on broadly. But HIV gave everyone the sharp focus, as Joan mentioned earlier. It was pretty remarkable from the first method in '84 to identify an AZT's activity in '85, and it being approved by FDA in '87 was just an indication of how important and how really scary this disease was at that time. The team that did this have their names on them here. Mitch Mitsuya, who spoke yesterday, Bob Yarchland, guy in charge of the clinical studies, and Sam Roeder, who was director of the National Cancer Institute. And this is the HIV version of this meeting, and this was held in San Diego in 2012. You can see Raymond and I are in the picture too, and you can see at the back it says HIV DART on the chalkboard. Doug Richmond took this picture, and this is, at the time, was his son's restaurant. And it was a wonderful event and a good time for all us to come together and talk about the work we've been doing over the years. So after AZT, the series of dideoxynucleosides were approved, DDI, DDC, and D4T. DDI and DDC also came out of Sam and Mitch's NCI lab, and I got to know them quite well because at Bristol Myers we licensed DDI and took that into development, and that turned out to be a close collaboration over a number of years. At a similar time, we were working with Bill Prodrugs at Yale, and eventually came up with D4T would be the product to pursue. I'll never forget sitting in a conference room by my office, and everyone was talking about D4C. It was more potent, like DDC is very potent. And Ismail Ghazuli said, we shouldn't overlook D4T. And it was just one of those moments that focused all our attention directly on D4T because, like DDC, D4C was substantially toxic, and it also was very unstable. It was easily hydrolyzed. So that moment, we switched on to D4T, and the approval of D4T followed AZT about 7 years, but those two drugs were the mainstay of antiviral therapy all through the 90s. So patients either on D4T, AZT, and eventually two other drugs, and they were just known as the thymidine analogs. In this push to come up with new drugs for HIV, people are pulling down off the shelf or resynthesizing compounds that are already known in the literature and testing them. And the key was to have access to an assay. So I mentioned already that Mitsuya developed the early assay at NIH. Raymond Shinazi, who worked for Bill Prusoff in the 70s and had long since moved to Emory, also developed an HIV assay at Emory University. So when Taishan Lin resynthesized D4T and Prusoff's group, it was sent to Regimen, and so there's the very first D4T publication. In parallel, two other groups were working on it. Masanori Babas was with Eric de Klerk's group in Belgium, and Hamamoto is in a Japanese group in Japan. And so these three publications came out in the same year, and it results in different versions of history. And so there was a version of the history in New Haven, and there's a version of the history in Leuven, Belgium. So Mick Hitchcock, who also worked in the Bristol-Myers group that was involved in the development of D4T, in 2009, he and I thought we should write the definitive history and do it with Eric de Klerk and Bill Prusoff so that we'll have a publication that the two giants in the field will both be on, and it will be the definitive history because we're going to have to negotiate every word and every paragraph. And we got that. It is published in an Antiviral Research issue dedicated to antivirals in 2010. And we just really had a lot of fun. And I really enjoyed working on this review article and talking to Bill frequently by telephone from where I was in California. Just another aspect of sort of bringing home the point that if you want to be successful in drug discovery and development, you need to have chemists around that can help make that work, and so multidisciplinary teams are the key. We developed a variety of syntheses to allow DDI and D4T to be evaluated quickly in preclinical assays and to be manufactured. The first paper on there, Ernie Prisby was my colleague at Syntex, and it's kind of how things work in a circuitous way. We, as a sidelight, built a small laboratory in his garage and had a company that we named Biothon. And that was at the time when oligonucleotide chemistry became important, DNA synthesis and also DNA sequencing. So we developed the first published approach to make dideoxynucleosides because we were making dideoxynucleosides to sell to another company so they can make the triphosphates for Sanger sequencing. And so never did we realize that in '85 when we published this, that a couple years later we'd be developing DDI as a drug. The D4T, the Mansouri et al. Paper in JMedChem, that's where we broadly characterize the efficacy of D4T and quite a bit of pharmacology, and it's the work that we did to select D4T for development. Another thing that in working on the dideoxynucleosides, at Bristol-Myers, amikacin was made from a lactone that's dietized glutamic acid. So we were easily able, to go over to the plant with a bucket and take the starting material, and we made all the isomers of the dideoxynucleosides, all of DDC, and we found that L-DDC had very potent activity, and that eventually led to a series of L-nucleosides, which we already heard about that Raymond knows quite well. And lamivudine became, throughout the 90s and beyond, the second drug used with thymidine analogs. So you can think thymidine analog, lamivudine, and a third drug. So I'm going to switch now to nucleotide analogs, which is a big part of my history and in terms of how we built Gilead to have major antiviral therapies. Raymond mentioned ganciclovir, that's a Syntex compound, and again, with Ernie Prisby, we considered, what about making a stable analog of ganciclovir monophosphate? So at the top right, you see the monophosphated ganciclovir, and we just replaced the oxygen with a methylene. Phosphates hydrolyze phosphonates don't. And up until this point, it was thought nucleotides are too polar to get into cells and would not have antiviral activity. That turned out not to be correct. And the nucleotides previously made that didn't have antiviral activity, they got in cells fine, they just didn't inhibit the virus. And so ganciclovir phosphonate did get in, a little bit gets into cells, and it was just as potent against CMV as ganciclovir. And then the other thing we noticed is you can pre-treat cells with nucleotides. Replace the media. If it's a nucleoside, it comes out of the cell, and the cell can be infected. If it's a nucleotide, it stays in the cell, and the cells are protected from infection. And that important observation led us to focus over a number of years on coming up with potent and ultimately very selective nucleotide analogs. So around this time, I moved to Bristol-Myers, and you heard about the D4T and DDI. We started also collaborating with Eric de Klerk in Belgium and Tony Holy in Prague on phosphonate nucleotides. And in this case, for the chemists, you can see that tenofovir is a phosphonomethyl ether. And that oxygen in that side chain is very, very important because it's electron withdrawing and tunes the acidity of the phosphonate to be more like a phosphate, and that led to the broad class of nucleotide analogs with biological activity. This is a picture from 1987, and first of all, you can see Bill Pruzoff sort of in the middle, right above the corner of the swimming pool. Again, as Raymond said, he was with us all over the world. Eric de Klerk and Tony Holy are in the picture also, and when I negotiated an agreement with them on this collaboration, I put in the agreement that we have to meet, or at least I have to update them every 3 months. And so it wasn't really heavy duty to go to Italy and have a meeting with them. And we met typically at scientific conferences all over the world, and much like that I've met with Bill many times during this time frame too. Another person in the picture, Ron Borkart, is recently retired from the University of Kansas. I'll come back to him, but he typifies the type of medicinal chemist that comes to these types of conferences to interact with people across the scientific spectrum. So, in the case of, I just want to check something. Yeah. So, in terms of tenofovir and D4T, TDF is Viriad, we went head to head, and D4T, which had, as I said, been the mainstay of therapy, had very, very significant side effects related to mitochondrial dysfunction. And this is peripheral neuritis, neuropathy, lipodystrophy, and lactic acidosis. And TDF greatly reduced those side effects, and that's what led to the momentum of TDF being the mainstay of antiviral therapy for until this day, actually. Viriad was approved in 2001, and when this data came out in about 2004, we saw just a very rapid switch over of thymidine analogs to TDF. Another very interesting thing, the mitochondrial toxicities may be somewhat a little bit foreign in imagination, but this isn't. These were patients naive to AIDS therapy who started therapy once they had AIDS. And of course, everyone knows AIDS is a wasting disease. And going on therapy over the first 6 months, the patients in both arms' weight went up about 2 kilograms. But after that, the toxicity of D4T would start to kick in, and there would be some weight loss. Whereas on TDF, you saw continued weight gain. And this was the type of data that just is so straightforward to understand about what the toxicities of D4T were. So this is back to Ron Borkart. Ron says, Ron taught medicinal chemistry courses, of course, at University of Kansas, but also and many organizations around the world, including biotech and pharmaceutical companies. And he'd start off his lectures with, drug candidates aren't high-affinity ligands. They must have drug-like properties. And that sounds easy, but that's hard, and it's why it's very important to have chemists that are sort of at the professional athlete level involved in your programs. So tenofovir is shown at the top, and what we've had a long-term focus on is coming up with prodrugs that allow nucleotides to be orally administered. A drug like, or a molecule like tenofovir has to be administered systemically. You can't do it orally. And in fact, Henry Mazer, who spoke yesterday, did some of the very first IV studies for CMV and for HIV with our nucleotide analogs about 25 years ago at the NIH Clinical Center. So TDF, as I said, was approved in 2001. And along the way, we kept working on other prodrugs and came up with a separate prodrug, TAF, which is now Vimlidi for hepatitis B, that allows us to lower the dose of Viriad by a factor of 90%. And that reduces, again, the side effects. And so TAF is better tolerated in TDF in terms of kidney toxicity and bone marrow toxicity. And I'll talk about the products that came from this. At the time we launched Viriad at the end of 2001, we were in a situation of what do we do next for HIV? And it was kind of clear at that point that the big advance would be to come up with single tablet regimen where 3 drugs are in 1 pill given once a day, not twice a day, but only once a day. And that improves compliance and reduces resistance. But the important thing it does is you take all or nothing. If you take that 1 pill, you've taken your drugs. If you don't take it, you haven't taken any of them. And it's the partial regimen that was giving rise to resistance and leading to treatment failure. So we embarked on that immediately, and we also recognize eventually if you can get enough people treated, viral load in the community will be down and you'll reduce transmission. So the obvious place to look was at Raymond Schnazi again, right? And the 3TC was the established product on the market, and a company in North Carolina, Triangle Pharmaceuticals, was developing emtricitabine. And it was really fortuitous that we were able to combine that effort with the Viriad effort to come up with Truvada and have the at that point, 2 drugs in a single pill. Then the third product is pretty clear that efavirenz would be the best to combine because efavirenz has a long half-life like FTC and TDF. I want to make one other point on FTC. FTC was, what happened is Dennis Liotta, Raymond's colleague at Emory University, developed a synthesis of how to make these types of thioxylanes attached to a nucleoside in a highly stereoselective way. And that allowed the synthesis of a number of compounds to be evaluated. And having a fluorine on the ring may seem like a minor modification, and you wouldn't know which direction it would go. But it turned out it had higher affinity, the triphosphate had higher affinity for RT, and that means it's more potent against HIV-1. And another advantage it had is the longer half-life. And so that longer half-life made it the perfect pair of TDF to make Truvada, but then efavirenz with the longer half-life was also a desirable third agent. However, the efavirenz situation was complicated because it was shared by Bristol Myers Squibb and Merck in a variety of different ways in different territories. So it made it hard to come to an agreement, and it's still, to this day, I think, the best example of companies collaborating to bring through a new breakthrough product. At Merck, it was Per Wold-Olsen who led the effort, and at Bristol Myers, it was a fellow I mentioned before, Ismail Ghazouli, who raised his hand about D4T, He was in virology, and he had moved into business development for Bristol Myers, and he led the Bristol side of this. And so we combined 3 drugs into 1 pill. It sounds simple. It took us about 2 years to get it because we had to take 5 different formulations into the clinic until one was bioavailable. So it's just every step, there are multiple barriers to get there. But the single tablet regimen transformed HIV again. And you can see, going from the left, the third product in is Atripla, and that brings in efavirenz or Sestiva. And Truvada was also approved for PrEP in 2012. And we had launched 2 additional single tablet regimens in 2012, Stribild, which is L-Vitagavir, an integrase inhibitor, plus cobicistat, a booster, and Caplera, which is in collaboration with Johnson & Johnson for Ropivacaine. Then the TAF products are shown below. Gemvoya, which is like Stribild, but with TAF instead of TDF, was launched just 2 years ago. And Gemvoya, in its first year of launch, is the fastest ever rate of patients going on to an HIV agent. So it may surprise you, in 2015, so many years after AZT was approved in 1987, that this is the most rapid launch of AIDS drug in history. It's, of course, there's a much larger number of people being treated now, and being on TDF for a long period of time, one worries more and more about the long-term toxicity, so there's a built-in patient pool to jump on that. Then we've launched 2 additional TAF products for HIV, and in February will be the PDUFA date for BF-TAF. Bictegravir is an integrase inhibitor. It doesn't require boosting. So in 1996, that was the first year that people could take 3 drugs for HIV and control the virus. The average survival was not that great. The average age of a person with AIDS was in their 30s. The problem with taking these many pills, as I talked about earlier, people can't take all these medications, so you end up taking a partial regimen, resistance developed, and people are running out of treatment options. But now, 20 years later, the average patient with HIV in this country is now over 50 years old, has been treated for many years. The average survival has gone from 19 years to 53 years post-diagnosis. And of course, that's a projection, but that's really remarkable in this period of time that at first in 1996 was the first time we had 3 drug regimens to manage the virus, and now 20 years later, people with HIV can live a normal, healthy life. Hugh Longo, I think, coined this. He certainly said it enough. Early treatment initiation to prevent transmission and preserve health. The theme of what people talk about for viral hepatitis or HIV is how do we identify people and get them into care, make sure they're virally suppressed or cured if it's hepatitis C. And that's how you can break the cycle of new infection. There's been quite a bit of work showing that we should treat, and now it's well accepted. Everyone, when they're diagnosed of HIV, don't wait, and make sure we diagnose more. And if they're on therapy, they will not transmit the virus. And also, I mentioned Truvada was approved for PrEP by FDA 5 years ago, and that has become, with a lot of controversy, it's become well accepted. And so HIV negative people who are at risk can take Truvada and protect themselves. So the global estimates now, as of the end of last year, is 36 million people. 1.8 million still became infected, so that's way, way too high, and 1 million died of AIDS-related illnesses. But now of the 36 million, about 20 million globally are being treated. And that's remarkable to think that we've come so far in the treatment of HIV that 20 million people around the world are receiving the benefits of therapy. Raymond talked about this. In 2003, we started thinking about how do we make Viriad available in lower income countries, and we started a program, but honestly, it didn't go anywhere. In 2006, we revamped it to do country-by-country registration. At one point, we thought if the WHO has guidelines, then people take the drug, but it just doesn't work that way. It needs to be approved country-by-country. We offered our branded products at no profit or discounted if for middle income. And then we took an innovative approach to intellectual property by transferring licenses non-exclusively to manufacturers in India. And the key was we would actually transfer our proprietary manufacturing technology so a company in India could start scaling it up and within 5 months have approval to sell the product around the world. If they tried to do it on their own without a transfer, it would maybe take 2 years before they could work out the process details. Then in 2011, we did Access 2.0, and retrospectively said 2006 was Access 1.0, and had all our learnings to figure out how to better manage the program. So you can see in 2006, there was a small amount of branded tenofovir in the lower income countries. But as the generic manufacturers came on, by 2011, it was starting to take off when we redid how we were thinking of a number of the logistics of this, working with partners all over the world. And as of last year, we're well above 10 million people on this product that is sort of stable at our brand at the bottom, but 90% of it is manufactured by a variety of manufacturers on a very, very large scale in India, and it's highly cost effective when it's made on that large scale. So the summary of what I talked about is in sort of over 30 years, there's been remarkable advances in the treatment of HIV, but with the launch of Viread TDF in 2001, things have progressed very rapidly. And we are eager to see the same thing progress in viral hepatitis. So when I talk about what we've done in the developing world, I wouldn't want people to get the impression that I think Gilead had a major role. There's many, many stakeholders that come together. Everyone knows that the Global Fund and PEPFAR over the last decade have done much to fund programs that not just drugs, the vast majority of the effort is in medical care. FDA and WHO do the approval of these products so that we know they're manufactured to good quality. Various groups work to improve access. Clinical studies, we work with hundreds and hundreds of sites all over the world, and many other groups work independently to demonstrate the value of different types of therapies. And WHO guidelines, of course, are critical. So we've had a number of nucleotide antivirals for hepatitis B and hepatitis C. Hepcera was our first treatment that we had approved for hepatitis B, and that was approved, believe it or not, 15 years ago. It seems like yesterday to me. And it was a remarkable product. It's obsolete now. Its safety profile is not great, but it benefited a lot of people. And even back with Hepcera, we saw the number of patients waiting on the transplant list for liver transplant go down because of the benefits of Hepcera. That was superseded by Viread in 2008, and Viread's safety profile is so much better than Hepcera. It just, we didn't encourage anyone to stay on Hepcera once Viread was on the market. And with Viread, we were able to garner quite a bit of long-term data, 5 and 10-year data where we keep people in studies, and showed that liver fibrosis not only stops progressing, but can stabilize and in fact improve. And then just last year we launched Vimlidi, which is TAF for hepatitis B. The dose of Viread is 300 milligrams, Vimlidi is only 25. And that design of that prodrug for oral absorption with Vimlidi also enhances cellular uptake. And so we are able to achieve an antiviral effect similar to that of Gilead, but with a much lower dose, and as a result, it has a better safety profile. Then on the hepatitis C front, starting with Simplification, we've been able to launch a hepatitis C product every year. First, Hervoni, which includes an NS5A inhibitor, then Epclusa, which is pan-genotypic, then Vosevi. And the interesting thing about Vosevi having a nucleotide, an NS5A, and a protease inhibitor is this probably will have benefit to resistant people. Two people have been treated and have resistance and failed the regimen. These direct-active hepatitis C regimens are highly, highly efficacious, but as we treat millions of people, there'll be more and more failures that will need to be treated with something that they can still respond to. The other thing, this revolution just over the last 4 years has replaced the many, many years that those of you who are physicians in the room have been using interferon regimens to control on a limited basis hepatitis C because not too many people could tolerate it. I travel the world, as Rema was saying, to lower income countries, and people talk about let's try to get single-use syringes available so we reduce transmission of disease by not using syringes multiple times. From the point of view of a chemist, I think about Sovaldi, Hervoni, the like, has eliminated 48 injections when you replace PEG interferon. And so you just think, you have 1 million people, you just gotten rid of 48 million single-use syringes. And it's something we don't think about so much. I know the chemists, I'm sure, Ray, but you've thought about this. Early in our career, there were all these injectable antibiotics that have been replaced by oral medication. And it's one of the big advantages if you can convert something that is not oral into an oral regimen. It greatly increases access around the world. So for hepatitis C and HIV have different geographical distributions. There's a lot more people with hepatitis C in the world, but it can be cured. So for HIV, our access program is about 130 countries. For hepatitis C, it's about 100 countries. And it's pretty much the same thing. Our brand is offered at discounted prices. Our licensing partners can provide generics in the territories that are defined. And we have a lot of them. It's the same, it's full transfer technology. We don't inspect what they do. They're required to seek approval by appropriate regulatory bodies. We now have 11 manufacturers in India that can produce for these 101 countries, plus 3 in-country manufacturers in Egypt and Pakistan, and you can see the logos of the many partners. SOF-based regimens, so SOF was launched, not too long ago, and it's come up fast, and it's coming up very fast. In the patients we know treated with SOF, because it's either brand or licensed generic, it's now over 2 million people and continues to grow every quarter. So hepatitis C, we talked earlier about preserving health in HIV. DAAs do that for hepatitis C. This VA cohort goes back prior DAA. It includes patients who were treated with interferon and was just presented a few weeks ago at ASLD. The patients who aren't cirrhotic are less likely to progress to HCC. Those are the 2 lines at the top. But the ones who do not have SVR have a higher rate of hepatocellular carcinoma. Then cirrhosis, if hepatitis C goes to cirrhosis, that's a much worse predictor, but still SVR gives a better outcome. This is a very large 62,000 patient cohort. There's something else I want to talk about, and that is a difference of HIV and hepatitis C in terms of treatment duration and what that means for access. Hepatitis C, 71 million people need treatment. HIV, 20 million are on treatment. If you work your way down, because AIDS patients take a pill, at least one pill a day, every day of the year, that's over 7 billion pills in a year, and that's year after year. It'll be 7 billion this year, it'll be 7 billion next year, maybe it'll be 8 or 9 the following year as we treat more people. So that represents a colossal scale of manufacturing, and that is highly economical and provides manufacturers a lot of incentive because that's an ongoing business that they'll have for their foreseeable future. Hepatitis C, there are more patients, but some patients can be cured in less than 12 weeks, some take 24 weeks, but just assume one pill a day for 12 weeks. That's 6 billion pills total. So as we slowly cure people every year, and it's going to take a lot of work to diagnose, it's not going to come anywhere close to the business that is generated for manufacturers by HIV. And we heard yesterday the challenge with hepatitis C. Also, most patients aren't diagnosed, and the testing can cost more than the generic price. But that'd be 100x if you have to test 100 people to find one patient. So there are major, major challenges for hepatitis C that are different from HIV. Again, there are many stakeholders that have created the environment where so many people can be treated. Product procurement, clinical implementation, strategic plans for treatment, diagnosis, and prevention. It really takes the national health ministries, every single country that embarks on this to have a commitment to make it work. It's without the national commitment, it's very, very hard to have inroads. And so what we've seen in Egypt or the Republic of Georgia represents two countries that stepped up early to have a big national commitment. Treatment guidelines are important. And again, hepatitis C is, even within a country, isn't uniformly located across the population or geography. So it's very important to have the appropriate treatment guidelines in appropriate places. And additionally, the patient groups for viral hepatitis aren't as mature as what we've seen in HIV for decades. And patient groups are becoming increasingly important advocates for viral hepatitis. So with that, that summarizes what I want to talk about. I'm very, very pleased to be here to be reminiscing about Bill Prusoff today, and thank you very much for your attention.